I'm going to correct my introduction slightly, which is okay. I haven't spent the majority of my life in the boron industry. I work for Rio Tinto, which is the mining company that owns the boron mine in California. And I worked for them for almost 5 years. And that's the extent of my boron experience. But I can tell you, after that much time just concentrating on one micronutrient, I should be able to answer at least, half of the questions you guys have about boron. So the rest of my life started off on the farm in north central Iowa, and I farmed there for about 20 years after my dad passed away. And that was a great farming experience. I pretty much did what I set out to do in farming. And so I'm someone who you might perceive as likes to challenge myself in learning new things all the time. So I took the opportunity to move to Illinois for a while and got involved in feeding horses, which is wild. But I can give you insights into pretty much every horse barn in the Chicagoland area, if anyone ever needs that information. But there I went into retail ag, and I worked for a big co-op up in Wisconsin, ultimately worked for Winfield Solutions for quite a while. And then all of that experience was in precision ag. So I did a lot of precision ag work. So anything about the mechanical part of it, I did, but also then focused on data management. And so I realized, and I was talking to a gentleman here about data, when we were talking about soil or tissue samples, and, looked at just all of the aggregate data the Winfield Solutions collected around tissue samples, which is a lot, and came to realize that most of it has no value, with the exception of those folks who decide to manage them like their soil samples, which is do the same exact thing year after year for your specific crops, and then look at trends over time. But guess what? Just like your soil samples, if you don't sample it during the same time of year and in a consistent manner, your data has less value, right? So I would encourage you all, if you're doing any kind of tissue sampling, no matter what your agronomist recommends, because they will recommend 1,000 different ways or things, but the best way, just looking at only the data, is if you take a crop and take that tissue sample exactly by crop development based on heat units from your planting date. It's pretty straightforward. It takes a little discipline, right? You got to note your planting date, number one, but then number two, keep track of the specific heat units that are accumulating on that crop, and then decide. I don't think it makes a huge difference exactly when your growth stage or heat unit sampling time is, but just decide to take a very consistent time year after year after year, and you'll ultimately start getting data that's actually usable. Because otherwise, any random one tissue test is of very limited value because of the interaction of weather, time, and crop stage, right? So you've got three variables that you're unlikely to hit, the same time consistently. So you have to do what you can to be as consistent as possible. So hopefully that little insight is helpful from that discussion earlier about tissue sampling. So boron, I will answer any boron question you might have. Are there any that come to mind before I start talking about something else? That's just out of the blue. And I'm old, so I don't mind uncomfortable silence until someone breaks it. How late is too? After it's physiologically mature. It's the same answer for every crop, right? So whenever that is determined to be, and I thought we had some good insight onto that earlier, when the majority of the pods have senesced, right? Because that's the flower. I think that's the technical term for that, after the flower has achieved its goal. You're not probably going to get much done with boron after that. Any other questions about boron? Boron is kind of a sneaky little thing. It's a micronutrient. One of the things you should know about it is that it leaches. It's the only micronutrient I know that leaches. And some people don't think about that part of it. And, it's like when you don't have enough boron, you need it. The plant will actually do things physiologically that it wouldn't do otherwise when it's seeking boron. There's boron in every plant cell wall. So think about that. Every plant cell wall. So every time the plant needs to add another cell or wants to add another cell or needs to develop some other structure within it, it must have at least a minimum amount of boron to take that next step. So you can see how a leaching nutrient, just like sulfur or nitrogen, right? In certain crop conditions or certain times of the year, even through irrigation, right? I mean, if you're irrigating over the top, you could be washing boron away from where it's needed and not even really be aware of it. So it's something that needs to go on. I always like to say that if you think about your need for nitrogen in a crop, which over the, lunch here we were talking about the concept of nitrogen on soybeans even at specific times, or it could benefit it significantly. Anytime you're considering any kind of nitrogen application, you should think about boron. You may not need it in every time, but you should think about, should I apply boron? Because nitrogen and boron have a huge symbiotic relationship and they don't antagonize each other. So you can pretty much add boron to any kind of nitrogen application very easily. So the unfortunate part, which I've heard from a gentleman over here, is that boron, unlike other things, if you apply too much, it becomes toxic quite quickly. And pretty much every agronomist that I ever met when I was doing my job at Rio Tinto always had the boron story where they killed something with boron, which easy to do because you just need a little bit if you don't have any, but if you apply too much, you easily get into the toxicity range. So it's where we have to be thoughtful about how we go about making those boron applications. So any other questions that are spurred from that conversation around boron? We can get to the bottom of boron pretty quick. It's, if you don't have it, you need it. And you should be at least thinking about it. If you've never considered boron, you probably have areas of your farm that are deficient. And rarely, if ever, does anyone ever discover that I have way more than I need. It doesn't happen. So, there you go.
Would you recommend a soil test versus a tissue test for identifying if you have enough, or what are some visual cues that you don't have enough?
Yeah, I think that the insight that I gave about tissue testing applies to boron too, because you'll see trends over time. And so it's just, unfortunately, the data around tissue sampling shows that the methodology is the most important thing in how you go about doing tissue sampling to get real insights, okay? So having said that, without a doubt, the boron in tissue tests is a real good indicator, and it will give you trends over time. And what you'll find is that the crop need for boron, if you're doing the tissue sampling, as I say, you'll find that the crop demand for boron, or the times where you'll find the greatest deficiency, is during flowering. Okay? So, and the reason is because the highest concentration of boron in any given plant is in the flower. So those little structures, no matter what kind of plant it is, all plants have flowering structures, and all plants have, places where there's boron. So you can look at this plant. These little, I believe they're called anthers in there. Those have the highest concentration of boron. In a corn plant, it's the silks. Silks have the highest concentration of boron. You think about a silk. How long does the corn plant use a silk tube? Any guess? I mean, It's 3 days. He said, I don't know.
It's a few hours.
It's not long. It's like a very short time. But yet the highest concentration of boron is that in that structure that you need for a few hours, it determines whether or not you even get a yield. So that's how critical, boron becomes. But like I said, if you need to make sure you have adequate amount of boron during flowering. So if you want to concentrate or focus on, where and when should I look for boron deficiency in my tissue sampling, make sure you're setting a regimen that gives you an indication before flowering. And what happens is folks make applications and accidentally hit the sweet spot for boron applications, meaning right before flowering. And they can have just dramatic yield results one year, and then try and do the same thing the next year and see nothing. Or worse yet, go backwards, right? And very easy, because the timing of that application is critical to when the plant has the highest boron need. Sorry, long answer. But
So in a soil test, are there certain levels we need to maintain?
yeah. So we always say that if you don't have 1 part per million, if you have less than 1 part per million, you're deficient. And you probably should be doing some kind of soil application. So there's the number, 1 part per million. That's it. If you don't have at least 1, you should probably put some in the soil. And the soil is the best place to manage boron, because over 90%, something like 97% of the boron that a plant uptakes comes through the roots. So that means if you're making a foliar application, or if you think a foliar application is going to help on boron, you only have a 3% opportunity anyway. But like I said, where it really shines is if you happen to hit that perfect sweet spot of 3 hours, or right before the 3-hour period of when that corn crop is using that silking thing. And you can have like, boom, huge jump if you're deficient, right? So boron is one of those things that once you're sufficient, absolutely you will not get any return to another application beyond that. Unlike, right, potassium, which we still really don't ever understand, because we can always add more and get more, it seems. So, but boron is not like that. Yeah. Any other boron questions? Are we getting bored of boron? All right. Well, one thing I want to talk about beyond that is just a few years ago, and I'm always, interested in different camps of understanding. And so, I think everyone's aware of this wave or this way of thinking where they call it regenerative agriculture, okay? And these folks are kind of like, they're mostly just like against everything that's normal in cropping agriculture. I'm not sure why. I haven't quite examined their bias or understood it, but they seem to be that way. But what comes out of that is they're thinking like another way, right? Well, one of these folks who, and I don't perceive him to be like, totally radical in that way of thinking like some folks seem to be, but he's a PhD guy, and he's focused on what's going on in the roots of a plant. And he just is looking closer and closer and closer, and he's down to the microbial level of the movement of microbes within the roots of a plant. So they said I could sacrifice this guy here because, most plant roots look similar, right? They have main stems, and they also have, what we would call, on this one, here's a good example, root hairs, right? And so this guy looked at these root hairs and he was like, well, what's actually going on in a root hair, and why does that structure exist? Our traditional thinking is, I love this picture because it helps me in this talk. We say, the roots are heading for the nutrients, and the nutrients are being mineralized or somehow synthesized by either application, right? We're applying it and we always talk about, this in this way, the fertilizer is ready to be uptaken. It doesn't have to be broken down by microbes, right? I mean, it's in a readily available form. That's okay, but there's still microbial action that's happening in the soil, and it's necessary to break down organic matter and other things that are in the soil, like the rock part, right? There's microbes eating that stuff, and what comes out of them is what the plant uptakes in theory, right? So he's looking at this and he's saying, well, what is actually going on? Well, what he came to realize is that these root hairs do not have uptake. They actually expel microbes. So we talk about, we need more root hairs because we got to get more. And actually the root hairs are the opposite. The flow is going out of them. And he demonstrates it in videos and shows how the plant kind of pulsates almost as if it has a beating heart. And these microbes are spewed out from the ends of root hairs. And he's not shown anything that's going in the root hairs at all. The only thing, the place of interception where nutrients enter is in the main stems. And you can see the main stem through this, right? And there's plenty of main stems in a plant's root system. That's where they're intersected and things are going in. But what's going in? Okay, maybe if he got down to an even smaller, better microscope, whatever, he could see actual nutrient flow potentially. But what he's found is that it's microbes going in. And the microbes going in are the ones that the plant sent out. And if you check the DNA of those microbes, they are the same as the plant, generally speaking. So the plant comes with microbes on the seed, because he's found these microbes on the seed, and it develops its own colony of carnivorous microbes that go out, become, absorb nutrients, come back in through the main stems. The plant zaps it with an oxide, strips off its cover. There's this name for it. I forget what it is. Do you know? No. Yeah, the cell wall. It's beyond that. There's another name for it because it's an actual animal, right? And what it does is it devours the nutrients, then become part of the phloem, right, of the plant. And the nucleus of the microbe is preserved, nursed back to life, divides at least once, and sent out the root hair to go get more nutrients. That's what's happening inside plants. They're carnivores. At least I wouldn't call that a carnivore. They're eating little animals, right? But so when we talk about what we're feeding, the chemistry side of us says we're putting these chemicals in, the plants are using those chemicals, they're adding cell walls in the We're actually, I think, what's happening is we're feeding these microbes. These microbes are being farmed by plants. The plants are just farmers, right? Farming these little microbes. And the microbes are doing all the work of the nutrient transfer. What do you think? Think it's possible? I mean, this guy, he has all the knowledge. You can look him up. His name is Mr. James White. He's at Rutgers University. He refers to he tends to fill some of his talks, not fill, but he mentions, I don't really understand why these companies, charge these farmers for seed, and they don't want them to save them. Because if they develop their own seeds with the microbes that they have on the same variety year after year, they would actually be able to increase the yield potential of that variety. And he also talks about how they observe microbes changing the nature, the physiological nature of any given plant or variety, depending on how healthy the population of those microbes are. It's incredible stuff. I got a farm now. Trent knows, he's been up there. And I am going to take an entirely new approach of thinking about how to raise plants because of that. Because if I'm not focused entirely, literally, on these microbes that are farming the nutrients out of that substrate, which is the soil, I think I'm missing the whole point of how a plant operates. So, all that to say, think about it yourself. Think if it makes sense. Think of how what you're doing now could play into that. And the one thing I'm going to do, and I've discovered certain things already, and years ago, some things were destroying the microbes. Some applications of some things that I was doing on my farm, I know were killing the microbes. And so I stopped doing that. Now, that was just observation of, hey, if I do that, I don't really advance my yield from year to year, which is what I was all about farming, right? Next year, I want a bigger yield. I want a better yield. Sometimes weather didn't deliver that, and that's where, hopefully some irrigation can mitigate that, right? But beyond that, how do I keep advancing? And I think the way that microbes interact with roots is going to give me some great insight into how to do that. I hope you guys got a little bit out of this demonstration here. So any last questions? Otherwise, I'm done. You can go take a nap.
So to piggyback on that a little bit, one of the things that we're doing, we started it last year. When those microbes die and that process happens, they give off CO2. There's a CO2 respiration. And when you send a soil test into most of the newer labs that are doing soil health, one of the things you get back is you get a respiration score. They put it in a chamber and they watch your soil breathe. And based on how much CO2 your soil gives off, they tell you how healthy your soil is.
So they're estimating the number of microbes?
Right.
Or the concentration of them in some number.
Yep.
And that's the only way you know of now to quantify the microbial base of your soil.
There's other ways, but that's like a big picture.
Yeah.
Okay, that's like saying what's the total health of that soil. It's not individual strains or individual species or anything like that. So a year ago, one of the soil probe companies we're working with, called Soil Tech Wireless, has a little moisture device that they were attaching CO2 sensors to. And so this year we did it last year. We went out and we put these CO2 sensors out in the field. So instead of sending it to a lab and seeing, well, what is that respiration one time? I want to watch my respiration through the day. I want to watch it when I irrigate. I want to watch it when I fertigate. Mm-hmm. And so we had them out last year. We got them out kind of late in the season. And basically what we were doing last year is we had them at the ground level, we had them in the canopy, and we had them above the canopy. And all we really learned was we need them at the ground. So this year, and I didn't have enough of them out to really get a good comparison. So this year we have them on dry land, corn and soybeans, and we have them on irrigated corn and soybeans. And I have them on some places we're doing some pretty high management. Mm-hmm. And it's just, there's a huge difference in what the CO2 level is based on how that soil is managed. Mm-hmm. So I think there's some things to learn there.
Yeah, have you correlated any of the CO2 readings to yield?
Not yet.
Okay.
That's next.
So then the next thing is, do you know of any good ways to measure the microbial volume or health of your soil?
So
to measure it, to say this is a grade A, this is a B, this is a C, or any kind of scale.
So the sampling that we sent, the lab that we work with primarily is Next Level Ag out of South Dakota, and they have indices that show that. We've also tried the microbiometer, and so we've looked at those two different metrics and still don't fully understand everything we're seeing.
Yeah, I think it's a great opportunity, really.
Yep.
I mean, I just think there's so much to learn from what those microbes are doing and how they respond to the things that we do, to amend the soil, right?
One of the other synergies, to take this a step further and back with what I was talking about this morning, the number one mineral that soil microbes need
Yeah.
is calcium.
Yeah.
The number one mineral is calcium. So is the function of plant response to calcium a function of microbial activity that's increased, or are they actually, in other words, it's an indirect consequence that I put calcium out, I increase my soil biology, and because of that, I have higher yields. So all of those interactions that we don't maybe fully understand.
Yeah, I think we're just scratching the surface. I was telling someone earlier, anyway, I think it was Tim. I was saying, I really believe that once we understand the symbiotic relationships between the microbial activities in our soil and how the plants are actually farming these things, we will move to another whole plateau of yield.
Yep. And it also goes back to pH. What pH does the biological activity is at the highest?
Yeah.
And is that the reason that those nutrients are the most available at that pH?
Or the microbes are the most efficient at transporting the nutrients. Who knows? My guess is probably close to neutral because most things work better there. But just a guess. Anyway. I love farming. I hope you guys do too. Thanks for letting me chat with you a little bit.
You bet. Appreciate you coming.
Yep.
Thank you.